BLAST P analysis, and also its metrics. So today we continue with um, MSA, multiple sequence alignment, meaning that we compare more than more than two proteins. So the proteins may come from different species, different organism, and the purpose of doing this, comparing the same protein but from different organism or different species, is to identify the conservation of amino regions in certain parts of polypeptide chain. We want to see that whether this particular area of polypeptide chain remains the same in different organisms. From this MSA, multiple sequence alignment, we can see whether this area is conserved or not. If conserved, meaning that this particular area of polypeptide chain is important. It has a significant function in cellular processes. If it is not conserved as we compare between different species, that particular area of polypeptide chain may not be important, may not be significant to us. So we can skip that area of amino acid. One of the common bioinformatic tool to perform multiple sequence alignment is named cluster W, as shown in this XPC server, cluster W. But nowadays, I think we have quite many tools for performing MSA, multiple sequence alignment. One of the newer one is cluster omega, if I'm not mistaken. You can double check it. There might be some differences in their output, but the objective of all these tools is to align protein sequences from different organism or species to see whether amino acids are conserved or not during alignment process. Okay, we click cluster W in XPC server. Then this is the page that we will see in XPC server. For this query box, we need to paste protein sequences. It could be 3 protein sequences, 5, 10, up to 20 protein sequences. And usually we paste all these amino acid sequences in FASTA format, indicated by larger than symbol, followed by protein name or protein ID, and below that, there must be a complete sequence of amino acids. The first amino acid for any protein should be methionine. Okay, if not, that amino acid sequence is considered incomplete, not fully annotated. We can use default settings as shown here, like penalty value, gap penalty, opening gap penalty, extending gap penalty. We can just use the default values here. From cluster W analysis, we can see the alignment of proteins from different species. For example, Mus bacillus, it is mencik haiwan kecil untuk experiments. The same goes to Vetus novagicus. It is also experimental, animal, Homo sapiens, about us, cyanates, not sure the same goes to the others, I'm not sure. So the same protein is used, uh, obtained from different organisms, then we do alignment. From this alignment, we can see the dark region indicating conserved amino residues. As we align all these 5 protein sequences. Other regions uh, which are not in dark color indicating less conserved regions. So probably from this multiple sequence alignment, we can see a pattern of sequence that may correspond to a certain function of this protein. Okay, from this multiple sequence alignment, we can generate. I mean, using the software, we can generate a pattern of amino acid sequence that can explain certain biological function of this kind of protein. Huh? Taking protein kinase, for example. So here we compare protein kinase from 6 organisms. Then we find that in this protein kinase, there are quite many amino residues are conserved among them. So this may explain about the important function of this kinase in certain biological process. At least we know that kinase is involved in phosphorylation of other proteins in either serine residue, threonine residue, or tyrosine residue. So this MSA successfully revealed the conserved regions in protein kinases across different species. Okay, so this is to show the difference between aligned sequences and also unaligned sequences. At the beginning, we may get this kind of sequences, shorter sequences, longer sequences, and so on. And during multiple sequence analysis, all these protein sequences need to be arranged. The algorithm, the software will do this for us. And following the alignment process, you can see the conserved amino regions. And probably based on these conserved amino regions, we can identify the function of this protein. And we may also relate this protein to um, the other protein-protein interaction network based on these conserved amino regions. From cluster W analysis, we can see 4 symbols: star, double dot, single dot, and dash. And each represent uh, different meaning. For example, star symbol indicates identical residues. Double dot indicates highly similar amino residues and others. At the bottom of this multiple sequence alignment, we can see all these symbols. For example, M, methionine residue, which is in red color. At the bottom of this alignment, we can see star symbol. So this star symbol indicate 100% identity